Welcome everyone to the NPTEL course on Food Science and Technology. This course is instructed by Professor H.N. Mishra sir, who is the professor in the Department of Agriculture and Food Engineering, IIT Kharagpur, and I am Shikha Sharma. I'm a research scholar in the Department of Chemical Engineering, IIT Guwahati, and I am the TA of this course. So in this live interaction session, we'll be discussing about week 12. So the assignment deadline for week 12 is 15th. So please submit your assignment on time. So the topics that will be discussed in today's class, which includes week 12, is uh, like it includes mainly about the circular economy concept in the food industry. So it includes about the concept of circular economy, reducing the wastage, reusing and recycling, then sustainable sourcing and resource efficiency, food industry, byproducts processing, and the waste utilization and the valorization of the waste, then green processing and packaging. Then this module 12 includes basically 5 lecture videos from 56 to 60. So lecture 56 is about the circular economy, linear economy, and lecture 57 is about the green processing, byproducts, and the waste utilization, and 58 is about fruits and vegetables processing industry waste utilization. Then lecture 59 is about fish, meat, and poultry processing waste utilization, and lecture 60 is about dairy industry waste utilization. So I hope you have all gone through the 5 lecture videos delivered by Professor Mishra. So in this live interaction session, we'll be discussing mostly the theoretical based questions on week 12, and which will going to help to solve your assignment. If you have any doubt from previous class or this class, please feel free to ask. Okay, so let's begin with the today's session. So today's first question is about which of the following statements are correct with respect to the linear economic concept in food industry. So it includes 4 statements that it follows a take, make, dispose model, and then second is it encourages reusing and recycling of the products. Third is it causes resource depletion and environmental pollution, and fourth statement says it is sustainable in the long term. So we need to tell which statement is correct among these 4 statements about linear economy. Okay, so let's discuss the solution of this question. So basically, let's try to understand what is the concept of linear economy first. So linear economy basically follows a model of take, make, and dispose. So it talks about that we need to, in this concept, it is about like we are taking the resource, we are extracting the resource, and then we are using it uh, for the production. So we are taking the resource or extracting the resource, then we are mm, making something out of it, which includes its production, distribution, and the consumption, and then it is wasted and disposed of in the environment. So the resources are extracted, used, and discarded without reuse or recycle. So it will dump into the environment. So in this system, raw materials are basically extracted and then processed and then used to make the food products or different products. These products are then consumed and ultimately discarded as waste after their useful life is over. So they'll be the resources will be taken, some product will be made, and it will be consumed. After that, it will be disposed in the environment. So this model basically leads to the resource depletion and environmental pollution as natural resources are consumed rapidly and waste is going to accumulate and to overall it will harm the ecosystem. So it does not promote reuse or recycle concept of the food, of the products, which are the key feature of the circular economy. So this makes the circular economy different from the linear economy. So in linear economy, there is no concept of reusing or recycling of the product, whereas in circular economy, we basically we will the product which is having the end of the life, we're going to uh, reuse it, or we're going to recycle that particular product, whereas in this linear economy we're not going to do that. Additionally, it is unsustainable in the long term. So this concept is unsustainable in the long term because we are we are depleting the natural resources, and we are creating the burden on the environment. So because it creates wasteful, it has a wasteful nature, and it produce negative environmental impacts. So it is not recommended basically. So here in this question, statement 1 and 3 are correct, rest are false. So it follows a take, make, and dispose model, which we discussed. Yes, it's true. So it encourages reusing and recycling of the product, which is false. So basically, it in the circular economy, this concept is being followed, but here in this linear economy, this concept is not followed. Then third is it causes resource depletion and environmental pollution. So it is basically because we are disposing everything in the environment, so it causes the resource depletion and environmental pollution, which is this statement is true, third one. And fourth is it is sustainable in long term, which is false because it is unsustainable in the long term. So this is basically statement fourth is false. So only 1 and 3 are correct here. Okay, let's move to the next question. Match the circular agri food practices which are given in column 1 with the respective enabler in column 2. So in column 1 we have four uh, uh, circular agriculture economic practices, circular agri food practices in column 1, which are rotational grazing, then promotion or promoting of the regenerative agriculture, then third is reducing the low impact food cost, and fourth is minimizing the food waste. In column 2 we have the enablers, which are subsidized price and tax breaks. Then V is public procurement strategies for local food. Third is invests in food waste processing infrastructure. And D is farmers market and local food festivals. So we need to tell which one is correct here, which is matching to which agri food practice is matching to the respective enablers. Anyone who would like to answer this question? We know that circular economy concept is basically different from the linear economic concept. So here we are basically producing, we are first taking the resources from the environment, then we are producing something from for our use, and we consume it, and after that we're going to encourage the recycling process or reuse process. So the we can reduce the process waste also. So these three R's, reduce, reuse, and recycle concept, we are going to use in the circular economy. Okay, Shiva is saying C here. Okay, let's see the answer. So D is the correct answer here. So basically circular economy, this is a basically economic model which is designed to eliminate the waste and ensure the continuous use of the resources. So it operates on the principle of reuse, sharing, repairing, refurbing, refurbishing, remanufacturing, and recycling to establish a closed loop system. So in this, basically, it is designed to on the concept of reduce of the waste, recycle of the waste, and reuse of the if the product can be reused, so it can the things can be reused in the process. So this concept of circular economy tries to reduce the waste which is generated and the resource consumption, and also it tries to reduce the pollution and carbon emissions. So this concept is basically having different uh, other sub concepts in this, like there is the concept of rotational grazing. So rotational grazing is basically comes under regenerative agriculture and locally sourced things. So in this, what happens? It supports the local sustainable farming practices, which is often promoted through local markets and festivals that connects farmers with consumers. So basically, it uh, this concept of regenerative agriculture and locally sourced products, it includes many concepts like rotational grazing, agroforestry, and silvopasture. Then it includes multi-species cover crops and minimizing the tillage or no tillage. So it has different barriers and enabling policies. So it includes like farmers lack the financial resources required to invest in the transition and or need support to minimize the risk to their profitability. So basically, when the farmer tries to shift its agriculture practices to the more sustainable practices, the farmers may need funds or financial support to invest in the transition of the technology. And for that, uh, for that enabler is that it can the farmers can use the regenerative practices and local food in public procurement strategy. So farmers uh, can go for instead of completely shifting to the agriculture organic agriculture practices, it can go to the regenerative practices. And the second is farmers can have the lack of knowledge and experience in regenerative practices and difficulties assessing, retailing, and selling of the infrastructure. So enabler is that the innovation incubators can be created so that the farmers market and the local food festivals can be arranged with the emphasis on regenerative farms. So the farmer can easily shift. So this regenerative agriculture, it basically includes that soil should be keep the soil covered always. So minimize the soil disturbance and maximize the crop diversity so that all the crop benefits can be reached to the soil nutrition, and maintaining living root year round, and integrates the livestock also so that the manure can be get and the soil health can be attained properly. So the rotational grazing, it supports basically the local sustainable farming, which is basically promoting the local markets and the festivals which connects the farmer with the consumers. And second is public procurement programs often can be prioritized sustainable and locally sourced produce, and it encourages basically the regenerative farming practices. So here, this rotational grazing is basically linked to the farmers market and local food festival, so that the connection with the consumers can be more, or selling can be increased. This second, which is promoting regenerative agriculture, it is related to that public procurement strategies for local food. So if the all the farmers will go together and uh, or all the customers will go and purchase in the bulk amount, so if the public procurement strategies will be there, so it will going to promote this regenerative agriculture practices. Then, so the government subsidies and tax incentives, basically, when we talk about the second concept, which is low impact diets and packaging designed for circulating. So basically, in this, the practices includes that the producers can shift away from intensive farming practices and resource and land intensive biological products towards smaller scale production and other activities. So the farmers from intensive farming practices, in which more of the soil nutrition degradation is there, so it can shift towards uh, the multi-crop production or like that practices. So prioritizing the production and processing of the food with lower impact should be there on the uh, or lower impact should be there on the resources which we are using for producing any food product. So the retailers can use consumer design to shift consumers' choices towards the low impact, healthier diets. Also, the consumer can shift its choices towards those foods which need less resources to produce. So farmers can shift to those practices. So the barrier is that reluctance to change the diet and the low consumer knowledge of the food impact. So for that, consumer awareness campaign should be run, so that the diet practices can be changed, and the price competitiveness of the low impact food is there. So the price regulation measure should be made, like it should be this, make like the government should promote like tax breaks or subsidized prices for this low impact food, so that it can be promoted. So the government subsidies and tax incentives we will need to lower the cost of the sustainable and low impact foods, making them more accessible to the consumer. So if the tax is low on those foods, or if the like the low impact foods, if the cost is low, or if there is subsidy on that, so it will be easier to promote these type of foods. So reducing low impact food cost is related to the subsidized price and tax breaks. Then eliminating the food loss and waste and creating value from byproducts. So best practices that we can implement this industrial symbiosis in food processing facilities, and the donation of the unsold goods can be there in store, price promotion for the things which are getting perish, so the things which are getting spoiled, so their price should be low. So the barriers in this is that lack of data on food loss and waste reduction. So for that, investment in collecting and monitoring the food waste data should be there. We should invest in such techniques which can collect the data regarding the food waste to quantify the what are the economic values lying in the food waste and how we can prevent. For these, we need to have some data. And second is that lack of infrastructure and markets for surplus food. So we need to invest in the infrastructures which can reduce the food waste. And also if there is food waste, so there can be technologies like composting and anaerobic digestion which can help to generate some kind of energy or bioenergy from those kind of food. So food is basically wasted, and it can after that, even if the food is wasted, we should try to reduce the food waste as low as possible. We should reduce it. After that, the food is wasted, the food waste can be utilized in the compost or to produce another kind of energy, to get extract some kind of energy, so that it will be helpful. So we need to like develop facilities for composting, biogas production, and food recovery programs to reduce the food waste. The fourth, which is minimizing the food waste, is related to investment in food waste processing infrastructure, which is C here. So answer D is correct here. Okay, let's discuss this question now. So based on the following depiction, identify the life cycle assessment model applied in the food sector. So this is the raw material production, then food processing, then distribution, and then use. So which is, so we need to tell which model of LCA is applying in A, B, and C. Anyone who would like to answer this question? Shiva is saying D, which is this. A is related to cradle, then B is related to cradle to gate, and C is related to gate to gate. Okay, let's see. So answer B is correct here. So A is basically related to gate. Then B is related to this is related to cradle to gate, and C is related to cradle model. Okay, so let's discuss this question. So life cycle assessment, basically it is an analysis tool that is basically a science-based scientific analysis tool is there that analyze the environmental impact from a product, process, or service throughout its entire life cycle, from the raw material or raw resource extraction, then through the process production, distribution, its use, and finally disposed to the or recycle, disposed to the environment or recycle. So in the complete life cycle of a product, how much environmental impact it is basically creating, that is calculated using the help of LCA. So it basically provides an holistic view to identify the areas for improvement. So and and promote more sustainable practices by considering the complete cycle of the product, process, or the service. So when we are scientifically analyzing the complete life cycle of a product or any process or services, so we can understand, okay, where the environmental impact is more and where is the less, and then we can improve on the existing system. And if we if that particular system or particular uh, basically one gate or particular system is producing more of the waste, so we can try to improve on that, and we can adopt more sustainable practices so that the environmental impact can be reduced. So this tool basically helps it helps in the transition of the linear economy to the circular economy. So when we do the LC analysis, so it helps in uh, how we can reduce the life cycle assessment. So it promotes basically the circular economic concept, in which we have them minimizing the waste and maximizing the resource efficiency is our goal to do that, to do this. So LC basically it follows the international standards ISO 14040 and ISO 14044. So the overall framework of LC includes that we will have some goal, and we'll define the scope, we'll define the goal, and we'll define the scope, and then we will see the inventory. So inventory analysis will be there, then impact will be assessed, and interpretation is done. So this is the overall framework of LCA. So first is that we need to define the objectives and scope of the study. So first the objective is being formulated, like determining the purpose of the study, such as comparing two products or improving a process environmental performance. So first the goal will be decided, that what we need to achieve, and then the functional unit will see. So we'll define the measurement unit of the product services to be analyzed. Then system boundaries will be set up, like so it means that the delimit the life cycle stages included in the study, like from raw material extraction to the final disposal. And fourth is the assumption and limitations is need to be made. So assumptions need to be identified, and the limitations of the study will also be seen here. Next is the life cycle inventory analysis. So first the data will be collected, so the about the material use and energy consumption, the what are the emissions, how much is the waste is produced, and about the other resources which are associated with the different life cycle stages. Then mass and energy balance will be seen. So we will see the all the inputs and output flows. Uh, so is it, how is it, and is it the balanced one or not? Then the system modeling will be done using of the LCA software and the tools to model the process and calculate how much is the total input and the output flows. Then life cycle impact assessment will be done. So classification will be done, like the associated inputs and the output flows with the environmental impact category. So there will be different environmental impact categories, like CO2 emission, then greenhouse gases one, and other uh, categories are there. So the impact will be assessed and basically classified and then assessed. Then the characterization is there. So uh, in this, the quantification of each flow's contribution to environmental impact category using the specific categorization factors. So they'll be categorized, and the normalization and weighing will be there. So the different environmental impacts will be basically normalized, and weighing basically they will be assessed on a common basis, and then a particular number will be assigned, or a particular weightage is given, and that will be assessed. Then life cycle interpretation will be there. So the critical review, so the data will be reviewed and the methods will be identified to ensure the studies, qualities, and the consistency, and identification of the patterns and trends will be there. So in this, analyzing the results to identify key contributors of the environmental impact. So from this analyzing of the that assigned value or the relative assigned value, it will be easier to identify what are the key factors which are affecting the environment. So those will be identified, and recommendations. Then the development of the recommendation so that the environmental impacts can be reduced or the process can be improved or the product performance can be improved, that will be done and that decision will be made based on those recommendations. So this LCA model basically has the 5 phases, which means it includes the raw material extraction. Then basically this product, any product is having these 5 phases, which includes raw material extraction, then its manufacturing and processing, then transportation, and then usage and retails, and then finally is the waste disposal. So in this is the 5 stages of a product life cycle. So in this, the LCA model, we have the different LCA models like gate to gate model, cradle to gate model. Like these, we have different models. So gate to gate model, this model basically focuses on a single process or the stage within the production life cycle, analyzing input and output between the 2 points. Example, only processing or packaging of the food. So there are different stages in the uh, basically product life cycle. So if we talk about the gates, gates are the steps, like the basic steps, uh, or the one step will be the gate. Like ideation is a, suppose if I am making idea, then I'm doing planning, then development, testing, launching, and review of the product. So ideation is a one gate, planning is another gate. Or if I'm saying that product processing, suppose in production, raw material procurement, then production, and then it's packaging. So suppose packaging is a one gate, so that is gate to gate model. So from one gate to another gate, if we are focusing on the single process or the stage, then it means that it is gate to gate model. Then second is a cradle to, oh sorry, the disturbance. Yes, okay. Okay. So yeah, so then is a cradle to gate model. So in this, what happens is a this model covers the life cycle from the raw material extraction up to the point where the product leaves the factory. So it is from the raw material we are taking to the final product which is leaving the factory, which is the gate. So that is cradle to gate. So it includes the stages like agriculture, then processing and packaging. So it is commonly used for assessing the environmental impact of the food product up to the distribution stage. So in this diagram, we can see that cradle to gate is the that raw material extraction to when it is the product is leaving the factory gate. Then is cradle to cradle model. So cradle to cradle is a closed loop model, so which evaluates the entire life cycle from resource extraction, so raw material extraction, its manufacturing, its production. So here in after the production, it is gate. So this is from raw material to the product manufacturing is the cradle to gate. Then after this is usage stage, and then finally end of the life, and fine again if we are seeing that is cradle to cradle. So this is a closed loop where entire life cycle from the resource extraction to its disposal here, and then again its recycling or reuse of the material will be taken. So it ensures a circular process, sustainable circular process, which is essential for sustainable food system. Whereas cradle to grave model, it includes basically the full life cycle assessment from resource extraction to the use phase and disposal phase. So it will take the resources will be extracted and then uh, came to the gate, which is the company's gate. After that, what how much like the product is used, so it will take the count of the product is used until it is disposed to the environment, that is grave. So it will take into account until it is the product is disposed to the environment, that is cradle to grave approach. So if we see this question, in this basically this is talking about the food processing only, which includes the gate approach. So it is gate to gate approach. So it started from one gate and end to one gate. So that is gate to gate approach. Then second is from raw material to its distribution. That is cradle to gate model, because from raw material it is started. Then C is cradle to its until its use, which is cradle to cradle approach. I hope this question is clear. Let's see this question. Match the byproduct from the cereal processing in column 1 with its application in column 2. So in column 1, we have the different cereal processing byproducts: rice, bran, husk, wheat germ, and then brewer's spent grain. And in column 2, we have its the different applications like edible oil and plasticizers, then production of biochar and silica, then sources of tocopherols and phytosterols, and D is animal feed and functional food ingredients. So we need to match here column 1 with to column 2. Anyone who would like to answer this question? Okay, Shiva is saying C here. Let's see. No, I think it's the previous one's answer. I'm not sure. Okay, let's discuss this solution. I think Shiva is saying C. Okay, only one answer. So C is the correct answer here. So when we are processing the paddy to the polished rice, so we can have different stages like husk separation. So we can get the byproduct as a from paddy to get brown rice. There is the separation of husk. So husk as a byproduct we can get. Then from brown rice to get the polished rice, there are different stages. So first is the thickness and the grading stage, where blue tinged rice will be separated. So after that polishing is done, so when polishing is done, bran layer is removed from the uh, brown rice, and then the length grading is there, so broken rice will be separated. After that color sorting is there, so colored rice will be separated, and the polished rice only is used for the food. So there are these different byproducts produced. in the rice industry. So rice bran, basically it is a rich source of rice bran oil, which is used for cooking and industrial applications like plasticizers production due to its high content of oleic and linoleic acids. So this is basically the rice kernel. So in this, the outermost layer is a rice husk around 20%. After that, the bran is there, rice bran, which is around 8%, and then white rice is there, which is the 70%, and this germ is there, rice germ, which is around 2%. So this is basically the rice bran, and this is the rice husk. So rice bran composition is this, and rice husk is this. So we can see here the oil amount is very high in rice bran. So it can be basically used for the rice bran oil production. And rice husk is basically an outer layer covering of the rice grain, and it is commonly used in biochar production through pyrolysis process, and it is basically the valuable source of silica which is used in the construction and industrial application. So the mineral ash content is very high in if we see here in this rice husk, and this rice husk is basically being taken and it is burned at very high temperature, and the ash we can get if we are burning in the limited amount of oxygen condition, which is around less than 700 degrees Celsius, then biochar we can get. And if we are burning in the aqueous environment, then hydrochar is what we get. So, which is this ash is very high source of silica, and it is used in construction and different industrial applications. Uh, so here rice bran is basically used for edible oil and plasticizers production, and rice husk is basically used for biochar and silica production. Then wheat industry, if we see, so this wheat kernel is basically that is having this starchy part is known as the endosperm of the wheat. It has high amount of, basically this contains the most of the part, which is 80 to 85%, and it has protein, starch, and fibers. And this is the germ part of the rice, sorry, of the wheat kernel. So it is around 3%, and it is having the lipids, vitamin E, vitamin B, enzymes are there, different antioxidants are there, minerals are there, and plant sterols are there in this germ part. And then if we talk about the outer layer, so it has basically uh, alveol alveolar layer, and there are different other layer like testa, then inner pericarp, outer pericarp. So these are the different other layers which are there on the rice kernel, sorry, wheat kernel. So this outer layer, or the husk part is there, which is basically having the high amount of the dietary fibers, both the soluble and the insoluble fibers are there in these layers. So this wheat germ, this part is basically nutritional dense, and it is the byproduct which is rich in vitamin E, which is tocopherols, and phytosterols, so which is used in nutraceuticals and heart healthy products. And this is basically this germ which is used for oil production, or it is rich in vitamin E. Whereas this bran, which is the outer layer of the basically outer layer, which is basically protecting the wheat kernel. So it is the basically rich in fibers, minerals, vitamin B6, thiamine, folates, and vitamin E, and it is also rich in some phytochemicals, and it can be used in basically as a source of the antioxidants and phenolic compounds also. So this wheat germ, basically it is the rich source of the tocopherols and phytosterols. So if we see here, if we are processing the wheat, so to the wheat flour. So first we need to clean the wheat, then conditioning, then glistening, then grinding and sieving. So when the broken wheat will is grinded and sieved, the wheat bran will be removed separately and wheat germ will remove separately, which is having high content of the tocopherols and the phytosterols. And wheat bran is basically rich source of dietary fibers and other bioactive compounds. Then next is the brewer's spent grains. So it is basically a byproduct of the beer production. So barley is there. So from barley to beer, if we are processing, so we can get different byproducts. So from barley, it is being cleaned, then steeping is there, germination is there, then drying, then rootlets removal will be there, then barley malt is the product we get, and after that the barley malt is basically milled, and uh, after the milling, the mashing and filtration is there. From there, this brewer's spent grain will be separated, the hops will be added, and the boiling and cooling is done. After that, the fermentation is done with the help of the yeast, and the surplus yeast will be taken as a byproduct, and spent hops will also be removed as a byproduct, and the finally the maturation is there, filtration is there, and the pasteurization and packaging is there. Finally we get the beer. So in this industry we get three different byproducts, like the brewer's spent grains, then we get the spent hops and the surplus yeast. So this brewer's spent grain which we get, it is basically the rich source of the proteins and the fibers, and it is used for the animal feed and used in the functional foods for increasing the dietary fiber intake. So this brewer's spent grain, it is basically for the animal feed and functional food ingredients. So answer C is correct here. So this this basically this brewer's spent grain, it can also serve as a substrate for bioenergy production, like biogas or the bioethanol, and used for extracting the valuable compounds like protein, hydroxylates, and the antioxidants. And it is also used as a soil conditioners or also for the compost material. It is basically used as a uh, like used in different food products, like breads and the extruded products, so that the dietary fiber can be enhanced or the vitamin and minerals can be supplemented. Uh, then also it can also be used as a bioplastic material for plastic production. Also, it can be used as bioabsorbent for wastewater treatment and production of dietary supplements and the functional ingredients. Okay, let's discuss this question. So there are different processes. This is basically the chemical processes for rice bran when we are making the refining, or when we are refining the rice bran oil. So we do certain treatments like physical treatments and the chemical treatments. So here in this question, we need to tell which is the correct sequence of the chemical process of refining. Okay, anyone who would like to answer this question? Shiva is saying C here, which is this. Okay. Let's see the solution part. Actually, here B is the correct answer. So this is the basic chemical, the correct sequence of the chemical process. So basically, rice bran oil, it produced from the germ and the bran part of the rice kernel. So if we see here, this is the germ part and this is the bran part. So from this is we get the rice bran oil, and when it is being refined, we can do from the two process. One is a chemical process and one another is a physical process. So chemical process, we take the crude rice bran oil. First degumming is there, then neutralization, then bleaching, then deodorization, and finally we get the refined rice bran oil. And when we are discussing the physical process, so it includes basically first is the degumming process, after that is the bleaching process, and then dewaxing, and finally the physical refining and refined rice bran oil we get. So the chemical refining process of rice bran oil, where the crude rice bran oil, which is having 4% unsaponifiables, like unsaponifiables means the complex mixtures of the natural occurring antioxidant compounds such as vitamin E. Then second is the oryzanol. Then also like this is 4%, this unsaponifiable uh, compounds are there. Then 2 to 4% free fatty acids are there, and 88 to 89% of the neutral oil is there, the crude rice bran oil. So it is refined in these four processes. First is the when we are doing the chemical process, so degumming, neutralization, bleaching, and deodorization. So in the degumming process, basically what is happening, we can do from the water and the chemical treatment. So here, water or acid treatments is we apply to remove the phospholipids and the gums, enhancing the oil's stability. So alkali treatment basically eliminates 93 to 94.5% of this oryzanol, gamma original. So uh, from the this crude original crude oil, whereas the degumming and the dewaxing process, it only eliminates 1.1 and 5.9% of these compound. So acid or alkali treatment is basically alkali treatment is preferred, so that most of the degumming can be achieved effectively. Then is a neutralization process. So neutralization means from caustic soda treatment, the free fatty acids will be eliminated so that the rancidity of the oil can be prevented. Next is the bleaching process. So bleaching is done so that the decolorization of the pigments can be done. So diatomeous uh, earth absorbs pigments or the oxidation compounds or the impurities, so that the color and oil uh, can be improved for the oil. So it can mainly activated carbon or bleaching clays can be used for this. And then finally is the deodorization, which basically includes the vacuum steam distillation process. So it removes basically the volatile compounds and the odors, which so that the neutral taste and the aroma can be achieved. So the offensive scents, like which is produced from peroxides, aldehydes, and the ketones, they can be, or there can be different distinct tastes and the aromas, they can be removed in the particular step, so that the refined rice bran oil we can get and we can use in the cooking process or the industrial applications we can use. So this is the correct sequence of the rice bran oil. So B is the correct answer here. Then next is the process of turning agri-food waste into the renewable energy such as the biogas is known as anaerobic digestion, then composting, incineration, or fermentation. Which is the correct answer here? So Shiva is saying A, anaerobic digestion. So he's correct here. So anaerobic digestion basically is a process where organic waste materials like the food scraps or the agricultural residues or the animal manures, they'll be converted into biogas. So it is basically a renewable source of energy which is composed of methane and carbon dioxide. So anaerobic digestion basically occurs in the absence of oxygen, where basically microorganisms break down these organic matters into the biogas and digest it, which is a nutrition rich byproduct. So this method is used for the waste management and production of the renewable energy sources. So it can reduce the greenhouse gas emission and improve the sustainable energy generation uh, processes. So here if we see in the diagram, so on this side, on the left side, if we see anaerobic digestion system is there. So there is this inlet where the food waste is put in this tank, where the microorganisms will digest this food product and convert the organic matter into the biogas. So biogas will be stored in the tank and digestate will be taken, and it can be used as a manure in the agriculture land. The next is the basically here if we see the manure of the uh, animals or energy crops or lignocellulosic biomass or the food waste, the sewage sludges, they can be basically used as a substrate to produce the biogas. So they can be put into the biogas production, and biogas we can get for energy production and digest it for the nutrition recovery. So here anaerobic digestion is the correct answer. Next is the thermal decomposition process carried out in the absence of oxygen to produce biochar, which is known as gasification, pyrolysis, torrefaction, or hydrothermal carbonization. Anyone who would like to answer this question? Shiva is saying B here, which is pyrolysis. So let's see. Pyrolysis is the correct answer here. So the thermochemical conversion of waste biomass, which is the for the biochar production, if we see, so there are different processes are there of the like from the help of the heat, we can convert into different other products. So this waste, so we can use the pyrolysis, slow pyrolysis, and the fast, and then hydrothermal carbonization, gasification, torrefaction, flash carbonization. So there are different methods, and the by using different methods, this is a biochar yield and syngas production at different temperature and residence time they operate these. If we see here, the maximum amount of biochar is produced from this torrefaction method, and also hydrothermal carbonization, and gasification is comparatively less, and fast pyrolysis is comparatively producing less biochar. Also, if we talk about the syngas, so gasification and torrefaction process are the main for syngas production. So we were talking about pyrolysis. So pyrolysis is basically the thermal decomposition process where the temperature is very high, around 250 to 900 degrees Celsius, where what happens, the biomass is basically break down into biochar, bio oil, and syngas in the absence of oxygen. Basically, this process happen in the absence of oxygen. So the lignocellulosic components like cellulose, hemicellulose, and lignin basically undergo the reaction processes like depolymerization, fragmentation, and cross-linking at this specified high temperature, and there can be resulting of the different products. The solid is biochar, and liquid is bio oil, and gas is syngas. So these all lignocellulosic compounds like the cellulose, hemicellulose, or lignin, they will basically the fractions of these will be broken down, and these biochar, bio oil, and syngas can be obtained. The solid and gas product, which means the biochar and the bio oil, so basically, whereas the gaseous product can be carbon dioxide, carbon monoxide, hydrogen, and the syngas. So basically, if we are putting biomass in the uh, reactor, so what will happen? Fluidized, this is the fluidized bed reactor. So fluidized gas is coming. So here, the cyclone separator is there. Here, if we see the char burning is there, and from this, basically, three products we get, which is gas, bio oil, and this biochar. So here, pyrolysis is the correct answer, which is in the absence of oxygen, it produces biochar at higher temperatures. So in pyrolysis, if suppose if we are taking the cellulose, so cellulose is basically uh, there is this mechanism how it decomposes. So cellulose is converted into active cellulose and char and H2O. So this active cellulose is basically it is again converted into this primary vapors, secondary gases, and char plus H2O. Finally, the secondary tar will produce. And in hemicellulose, the volatile compounds and the intermediate products are produced. These intermediate products are further converted into volatile compounds and char. Lignin is converted into gas plus char. Lignin can also be converted into tar, and lignin can be converted into gas plus reactive vapors. So this way, these cellulose, hemicellulose, and lignin will be broken down and produce the char. So pyrolysis is basically used for converting this agriculture waste, wood chip, and other organic materials into the biochar. Biochar is basically a carbon-rich, stable material which is used for soil improvement process and carbon sequestration. So the other process like gasification, it is basically in the occurs with the limited oxygen to produce syngas. So it is not in the absence of oxygen. Then torrefaction is basically the mild heating which is enhancing the biomass properties, whereas the hydrothermal carbonization includes basically the decomposition in the water which is at under high pressure. So pyrolysis is only the for the production of the biochar through high temperature heating like 300 to 700 without the oxygen. So here pyrolysis is the correct answer. So here this pole pyrolysis flow diagram is given that from waste how it is converted into basically the power, or it is converted into syngas, bio oil, and the ash or the char. Next question is on the char. So which among the following statement is correct with respect to the char? Like first is biochar has a high moisture content. Then hydrochar is produced under dry conditions. Third is biochar results from pyrolysis, while hydrochar results from hydrothermal carbonization. Fourth is hydrochar is formed only at low temperature. So we need to tell which statement is correct here. So here Shiva is saying C is the correct answer. Let's see. So he's correct. As we know that from the uh, organic waste or the agriculture waste, we can get biochar, ash, hydrochar when we are maintaining different kind of conditions. So agriculture waste, it can produce biochar, bio oil and syngas basically when we are doing the thermochemical technologies like pyrolysis or gasification, and it produce basically the biocrude, hydrochar, syngas, and hydrogen when we are doing hydrothermal technologies like carbonization, liquefaction, or supercritical gasification. And if we are using the biological technologies like the fermentation, digestion, and composting, it produces biogas, fertilizers, and hydrocarbon, hydrogens. And this biochar is basically having the low moisture content because it is produced through pyrolysis process, so which removes moisture and volatile compounds because it is operated at very high temperature around 300 to 700 temperature. And this hydrochar is basically produced through hydrothermal carbonization, which occurs in basically the wet environment at very high pressure, which is around, like, temperature can be around 100 to 250 or 180 to 250, but it will not take dry condition. It is using the wet condition and the high pressure, which is around 2 to 22 megapascal, to produce this hydrochar. Biochar is basically produced from pyrolysis process. Thermal decomposition is there without the presence of the oxygen, whereas this biochar is produced from hydrothermal carbonization, which uses hot compressed water for biomass conversion. And hydrochar is basically can be produced at both low and moderate temperatures like 180 to 250 degrees Celsius, but the temperature range can vary depending on the process and the desired properties. So only three is correct here, that the biochar results from the pyrolysis and hydrochar from the hydrothermal carbonization. And it doesn't just operate at the low temperature, it can also operate at the higher temperatures. And this is hydrochar produced in the wet conditions, and so it is wrong. And biochar is basically having the low water content because it is operated at very high temperature. So let's discuss this question now. Column 1 is having the byproducts from cereal processing like anaerobic digestion, then different basically in column 1 we have different processes like anaerobic digestion, torrefaction, and gasification and transesterification. And in column 2 we have the products which we get, which is biogas and digestate. Second is biopole, which is torrefied biomass, and C is biodiesel and glycerol, and fourth is syngas and biochar. So in column 1 we have different processes to convert the cereal processing waste or the byproducts, and in column 2 we have what are the different products we get from these applying these technologies. Anyone who would like to answer this question? Shiva is saying C, so she's correct basically. So basically anaerobic digestion we discussed already, that it is a biological process which breaks down the organic matter into the different, like, biogas, which is a mixture of methane and CO2, and other is digestate, which is rich source of the nutrition rich source of the rich source, so it can be used as a fertilizer in the agriculture field, and this process basically takes place in the absence of oxygen. Then next is the torrefaction, basically, which is a mild heat treatment around 200 to 300 degrees Celsius of the biomass at low oxygen conditions or nitrogen or inert atmosphere. So it produces basically the bio coal or torrefied biomass, which can improve energy density and hydrophobic properties, which is making suitable for the fuel. So torrefaction can be dry torrefaction, steam, and wet. So dry torrefaction, it can be oxidative and non-oxidative, and it can operate at very high, at moderate temperature around 200 to 300, and around time around 5 to 24 to 50 minutes, or basically. So this is in minutes. This steam torrefaction, it can operate at a temperature around less than 260 degrees Celsius for 10 minutes. And wet torrefaction can be operate in the presence of water or dilute acid. So it can operate around 180 to 260 degrees Celsius, around 5 to 40 minutes. And this next is a gasification process. Gasification means that we are converting the biomass into the syngas mostly. Syngas is basically a mixture of hydrogen, carbon monoxide, and methane, and also in the biochar through a high temperature process, like it has basically 700 to 1000 degrees Celsius, and limited amount of oxygen is there, and the two products we get basically the syngas and the biochar. So syngas basically can be used for power generation, whereas this biochar can be used for soil improvement. So it is basically a thermochemical, a partial oxidation process basically. So it is basically a thermochemical partial oxidation process where biomass is basically converted to the gaseous mixture like the carbon dioxide, carbon monoxide, methane, hydrogen, and small quantities of higher hydrocarbons by supplying a controlled amount of the oxygen or oxidizing agent at very high temperature. So there is high temperature, which is around 700 to 1000. Then there is a limited amount of oxygen, and this biomass is basically provided this much of heat. So syngas will produce, and also the biochar will also produced. So the producer gas will have the calorific value of around 950 to 1200 kilocalories per cubic meter, and the producer gas, or the this producer gas is basically the syngas only. So the producer gas contains basically the carbon monoxide, which is 13 to 19%, then hydrogen around 8 to 18 to 22%, carbon dioxide, which is 9 to 12%, nitrogen, which is around 45 to 55%, then methane around 1 to 5%, and water vapor around 4%. The thermal efficiency of this gasification, it depends on the moisture content of the feed material, and suitable moisture content is around less than 15%. Hydrogen production increases with increasing the process temperature. So this is basically the gasification process. So this is the feed stock, and there the biomass is dried. Then the biomass at high temperature, it is decomposed. The volatile compounds will remove, and the combustion reactions will take place. Then the biochar is separated using cyclone separator. Then syngas is separated differently, and biochar here is separated differently. So syngas is purified, and water gas shifting is there to produce the hydrogen gas, and H2 is basically, hydrogen is basically purified, and hydrogen we get from the this gasification process. Next is the trans-esterification process, which is a chemical process where the vegetable oils or animal fats, which is having the triglycerides, they're going to react with the alcohol or the methanol in the presence of the any catalyst, so that the biodiesel can be produced and glycerol can be produced. So this biodiesel is basically used as a renewable fuel, and the glycerol is used in the pharmaceutical industries and the cosmetic industries. This glycerol. So here in this question, if we see, anaerobic digestion is basically related to this biogas and digestate. Then torrefaction is related to biocoal or torrefied biomass. Gasification is related to biodiesel, sorry, syngas and biochar. And trans-esterification is related to biodiesel and glycerol. Okay, let's discuss this question. The microorganisms generally uh, used for citric acid production from fruit waste is Saccharomyces cerevisiae, Aspergillus niger, E. coli, and Zymomonas mobilis, which is used for citric acid production from fruits waste. Which microorganism? C. Zymomonas mobilis. Here answer is Aspergillus niger. So basically, this Aspergillus niger is the most commonly used microorganisms which are used for the citric acid production from the fruit waste. So Aspergillus niger is basically a filamentous fungus, is known for its high yield of citric acid through submerged or solid state fermentation. So it basically converts the sugar from the fruit peels, pulp, and other industrial, agro-industrial residues to citric acid, and make it is a very effective and sustainable process. Whereas this Saccharomyces cerevisiae, it is basically used for the ethanol production, and this Zymomonas mobilis is known for its role in bioethanol fermentation, and this E. coli is basically used in the recombinant technology. So basically, recombinant technology means where, like, suppose the DNA molecule is basically from DNA molecule formed from the laboratory methods for gene gene recombination, that will going to bring two different or the other different genetic material from the multiple sources, and creating the sequence that would not otherwise be found in the genome. So this E. coli is basically used for the this technology. Like, suppose if the gene of one microorganism is good, and the and it is having high production of, suppose, citric acid or any bioethanol, so that the this E. coli can be used in that, but transferring of the gene and producing the different genome material or genetic material. So here Aspergillus niger is the correct answer. So if we see this citric acid production, so the fruit waste is basically pressed and the juice is removed. Then chopping and grinding of the waste is done. pH and moisture is adjusted, and the fermentation is there with the help of Aspergillus niger. So the fermentation mash will be there, and from that the citric acid is extracted, centrifuged, and clarified, and the mycelium will be removed separately. Okay, let's discuss this question. We have this different this missing step is there. in the process flowchart of bioethanol from fruit waste. So if from fruit waste we are getting bioethanol, so from feedstock is there and it is being crushed, hot water is added, and the pre-treatment like physical, chemical, uh, physiochemical and biochemical processes are treatments are done. Then hydrolysis is there to convert the fiber into the sugars, like acid hydrolysis or enzymatic hydrolysis can be done. And then the Saccharomyces cerevisiae 10% it is added for the bioethanol production. So this step we need to tell which step is there. Then distillation is there, dehydration, and finally the bioethanol production is there. We need to tell here in red which step is missing. Anyone who would like to answer this question? Okay. So fermentation is the one. So here fermentation is missing. So fermentation for 72 hours at 40 degrees Celsius should be there here. So uh, by the help of Saccharomyces cerevisiae, uh, so fermentation is done, then distillation is done, and dehydration, and finally the bioethanol is produced. So basically this bioethanol can be produced from sugars, starches, and cellulose. It is basically convert from different pre-treatments, then hydrolysis and fermentation, then distillation and dehydration, we can get bioethanol. So the fermentation is basically the key step in bioethanol production, where the sugars basically are being hydrolyzed to convert into the ethanol by microorganisms like Saccharomyces cerevisiae, 10% volume by volume. So in this process, basically the glucose or the fructose, they are obtained from hydrolysis of the fruit waste. They are metabolized and the ethanol and CO2 will be produced under anaerobic conditions. So the conversion of sugar to ethanol is there here, which is later purified through distillation and dehydrated to produce bioethanol. And the anaerobic digestion is done for composting. So the oxygen involves breakdown is there. So uh, then extraction is there or a separation is there. So that is basically in the composting. This aerobic digestion is in the composting, whereas in anaerobic digestion is in the fermentation process. So the different parameters like pH, saccharification time, fermentation temperature, inoculum size, uh, moisture content to solid ratio, solid to liquid ratio, and the fermentation time, they all are basically going to affect the how much is the bioethanol is produced or bioethanol fermentation. So these all parameters going to affect the productivity. Then let's see this question that we have the extraction techniques like sonication assisted extraction or microwave, pulse electric field, and the enzyme assisted. And in column 2, we have the different the principle of operation, and we need to match this. Anyone who would like to answer this question? This is like very simple question. Okay, so answer is D here. So sonication assisted extraction, basically this technology uses ultrasonic waves around 20 kilohertz to 100 megahertz so that the cavitation bubbles can be created. The small bubbles will enlarge with the time, and they'll basically going to one time, after some time it will be so big that it will collapse and cause the mechanical disruption of the cell wall, so that the bioactive compound will be extracted easily, and like the catechins from the apple polymerase, they can be extracted easily. Like this is the ultrasonic probe, and it is there in the product, or the sample solution. So it can going to create the cavitation and enhance the extraction of the bioactive compounds. So if the direct product is in the contact of the probe, it will be direct, and if indirect is in the when we are giving ultrasonic treatment in the ultrasonic path, so that will be an indirect treatment. The next is the microwave assisted extraction. So this method basically uses microwave energy in the form of electromagnetic energy around 50 to 300 gigahertz to generate rapid heating, so which increases the solubility and diffusibility of the target compounds such as phenolics from the citrus peel. So it basically this microwave works in the two principle. One is the dipole moment. So water will act as a dipole, so that will be there, dipole moment, and ionic interaction. So there will be plus charges and minus charges. So they will basically, when the electric field will be there, alternating electric field will be there. So they will, these polar molecules and the charged ions, they will going to basically align themselves as per the whatever is the electric field is there. So based on this heating is there, and the uh, extraction of the phenolic compounds are there. So this is basically the microwave setup, where microwaves are coming from this in this sample. Then is the pulse electric field extraction. So in this, basically high voltage electric pulse will be there, so that the electroporation can be done. So this particular principle we discussed in the previous lectures about pulse, the principle of pulse electric field. So how the electroporation is there, and how it will disrupt the cell membranes and enable the efficient extraction of the polyphenols from the citrus peel. So this is the basically pulse electric field, where the electric field is applied so that the electroporation can be done, and the compounds which are present inside the cell, like the bioactive compounds, they can be extracted. Next is the enzyme assisted extraction. So the biological extraction process, like the enzymes, like the cellulose and the pectinase, they're going to break down the plant cells, cell wall, like the polysaccharides, and increase the permeability, and facilitates the release of the polyphenols from the any pomace, grape pomace, like that. So if we see here is the example. So in the spinach, basically unsold spinach is there. So the different enzyme mix, like the cellulose 40%, xylanase 41%, and polygalacturonase 19% is put, and the enzyme assisted extraction is done, and the extracts are being characterized for their bioactivity. Bioactive extraction, how much is the bioactive extraction is there. So if we see here, so sonication assisted extraction basically uses the cavitation from high frequency sound waves around 20 kilohertz to 100 megahertz to rupture cell wall, then enhance the release of the catechin from the apple pomace. And second is a microwave assisted extraction. So it employs basically the electromagnetic field around 50 to 300 gigahertz to create rapid heating, and pressure changes facilitates phenolic extraction from citrus peel. Then pulse electric field extraction, it applies short, high voltage pulses around 100 to 300 watt per centimeter that causes basically the electroporation and enables the polyphenols extraction from citrus peel with 75% yield. And fourth, which is enzyme assisted extraction. It uses cellulose and the pectins to hydrolyze the cell wall components, which increase the permeability for 98.1% of the phenols recovery or polyphenols recovery from the grape pomace. So here answer D is correct. Let's discuss this question on nanocellulose. So nanocellulose is widely used in biodegradable packaging due to its eco-friendly nature. Reason is that nanocellulose derived from cellulose has a higher surface area, excellent mechanical strength, and barrier properties, making it suitable for sustainable packaging solutions. So, which is the correct answer here? Anyone who would like to answer this question? Anyone who would like to answer this question? Okay, so answer A is correct here that both the assertion and the reason are correct, and reason is a correct explanation of the assertion. So nanocellulose basically, it is basically we see the from the fruit and vegetable waste. So the fibers are being extracted, the pulp fibers are being extracted, and from three process we can get the nanocellulose. Like when we do the physical treatment like homogenization, we can get nanocellulose fibers, and after that the chem mechanical process we can do and we can get the nanofibers. And if we further if we do the ultrasound centrifuge and dialysis, we can get the cellulose nanocrystals, nanocellulose crystals. Or from the nanocellulose fibers, if we do acid hydrolysis, we can get nanocellulose crystals. And then from biological process, if we talk about, we can go for the fermentation. So from the fermentation, then separation and purification, we can get bacterial nanocellulose. And then from the help of the from the pulp, if we do certain treatments like hydrolysis, alkaline hydrolysis, or ionic uh, liquid pre-treatment, we can get this nanocellulose crystals. So here, this is the wood. From this, there are different in this there are this porous structure, and in this cellulose bundles are there. So they are basically attached with this is the molecular structure and attached with the hydrogen bond. So these cellulose basically these are the having the crystalline and amorphous regions, and they can undergo the acid hydrolysis and enzymatic hydrolysis. Then we can get cellulose nanocrystals. So this is basically the structure of cellulose nanofibers. Then B, which is the structure of cellulose nanocrystals, and C is the structure of bacterial nanocellulose. So this nanocellulose basically is used in the biodegradable packaging due to its sustainable and renewable nature. And the reason basically appropriately explains this because nanocellulose basically derived from the plant. Cellulose has basically high tensile strength like the steel on a weight basis, and it is also having the excellent barrier properties like the resisting oxygen, grease, and moisture, and has the large surface area because it is in the form of nanoparticles. So it enhances its compatibility with bio-based polymers. So these can be ideally used for eco-friendly and biodegradable packaging materials. So here assertion is also correct that it is used for nano-cellulose is used for biodegradable packaging materials due to its eco-friendly nature, and also this nano-cellulose basically has high surface area and excellent mechanical property, barrier property, and that's why it is suitable for packaging solutions. The next question is regarding the fish waste. So fish waste basically it is converted like it is treated and it is converted into oil from the fish waste, and then different treatments are there, and some uh, like what is we need to tell which stage is this, and after that we get the biodiesel. So is it degumming, trans-esterification, acetylation, or gasification step? Anyone who would like to answer this question? Okay, let's see the answer. So trans-esterification is this the step. So fish waste is basically the oil is being extracted, and then trans-esterification, and then finally we get the biodiesel. So we already discussed somewhat about the trans-esterification process in the previous slides. Let's discuss again. So trans-esterification basically is this conversion of the oil into the biodiesel. So it is basically the chemical process. So when we have the triglycerides or the oil, we can mix it with the alcohol and the catalyst, and we can get the biodiesel and glycerol. So trans-esterification is a reaction where triglycerides from the fish oil react with methanol in the presence of the catalyst like sodium hydroxide and potassium hydroxide, so that biodiesel can be produced, like the fatty acid methyl esters can be produced and glycerols can be produced. Then trans-esterification of fish waste is basically in the presence of the acid like sulfuric acid and alkalis like potassium hydroxides as a catalyst. It is a catalyst. Then after that we heat it, and the fish waste is transferred in the biodiesel. So the process reduces the viscosity of the fish oil so that the biofuel we can use it, it can be suitable for the application as biofuel. So then degumming basically is a process of removing the impurity from the crude oil. Then acetylation is basically modifying the alcohols and the sugar, and gasification is conversion of the biomass into the syngas. So that is not biodiesel. So trans-esterification is the correct answer here. Now is the poultry blood, about the poultry blood. So poultry blood basically is a valuable raw material used for producing the blood meal, a nitrogen-rich organic fertilizer. Reason is blood meal contains high level of polysaccharides and complex carbohydrates, which enhances soil aeration and microbial activity, promoting plant growth. So which is the correct one here? Anyone would like to answer? Okay, let's discuss. So when we have this poultry, so for this poultry meat is produced from the live bird. So the slaughtering is done of the live blood. So the blood will come out as a byproduct, and then skinning and scalding is done. From that, feathers, heads, and the legs will come out. Then evisceration will be done. From that, the stomach or the internal organs like intestine, viscera, they will come out, and after that chopping is done and the carcass meat is we can get at the trimmings as the leftovers. is get as a byproduct. So the blood meal basically from the whole animal blood, basically, which is the blood, we can the from the whole blood of the animal, the centrifugation is done, and the red plasma is red blood can be separated from that, hemoglobin can be separated, and then if we are doing centrifugation, the plasma will have the protein, so we can get the proteins from this blood. And also from the whole blood, the coagulated blood will be taken, dewatering is done, and a dried blood meal can be obtained from here. So this blood meal basically is having very high source of the protein, around 85 to 90% is their protein, with nitrogen content up to 13%, making it a powerful organic fertilizer. So this is very high source of the protein. This plasma, the blood plasma, basically it is also a rich source of protein, which is having high amount of albumin like 63% and globulin around 3.5%, and fibrinogen is around 4%. So this plasma, basically plasma albumin, is basically used as a gelling and forming agent, and this thrombin basically it cleaves the this basically cleaves the fibrinogen and to give the protofibril, so it can be utilized also as an emulsifier. So also the polysaccharides and the carbohydrates, they are not there much in the blood meal, and the uh, they do not contribute to the nitrogen enrichment but may promote the this carbohydrates, they're going to promote the microbial activity in compost, not directly in the blood meal. So basically the poultry blood is a very good source of the nitrogen rich organic fertilizers because it is having high amount of the proteins. Also this blood meal is having the low amount of the polysaccharides. So this is assertion is true here, but the reason is false, and it is saying that it is having the complex carbohydrates and the polysaccharides of the high amount, which is false because it is not there, which enhances the soil aeration and microbial activity and promoting the plant growth. So this is wrong. Uh, so here assertion is true, but the reason is false. Okay, let's discuss this question. In column 1, we have the poultry and fish waste processing techniques like acid hydrolysis of fish scales, alkaline treatment of the poultry feathers, then enzymatic hydrolysis of the fish waste and rendering of the poultry fat. And in column 2, we have the respective products what we get from the after this treatment, like the biodiesel and the animal feed supplement, then fish protein hydroxylate for animal feed, and then collagen peptides for cosmetics, and keratin hydroxylates for bioplastics. Anyone who would like to answer this question? We need to match column 1 with column 2 here. Okay, let's see the solution here. So B is the correct answer here. So the byproducts recovered from animal and fish waste utilizes various hydrolysis and rendering techniques so that we can get the valuable products. So the acid hydrolysis of the fish scale will going to what it will going to do. So it will uh, going to break the scales, and so that the type 1 collagen we can extract, and it can be used in the cosmetic industry, basically also in the biomedical industries it can be used because it has a skin rejuvenating properties. So this is the fish waste. So from the fish waste, if we do the acid hydrolysis or the enzymatic hydrolysis, we can get the silage. So silage is having basically the heads around 9 to 12%, bones 9 to 15%, scales 5%, and viscera 12 to 18%. After that, blending is done with the grains, vitamins, and the minerals, and the pellets can be formed and it can be dried and packaged. So, the alkali treatment if we do for the poultry feathers. So this in the previous question here we discussed about how the poultry meat is basically from what are the different byproducts we get from the poultry. So the feathers we can get as we see here. So the feathers basically of the poultry, if we do the alkaline treatment, so the keratin can be dissolved and uh, we can get the keratin hydroxylate, which is a very helpful in the production of the biodegradable films and the bioplastics. So it is a basically sustainable material. And when we do the enzymatic hydrolysis of the fish waste, which is this, so it generates basically fish protein hydroxylates. So it is very rich source of bioactive peptides. So it can be used in animal feed, aquaculture also. So it has basically the collagen and gelatin as a high source of protein. Last, when we discuss about the rendering of the poultry fat, so it separates basically the fat from the poultry waste. So the fat can be used for the production of the biodiesel from the process of trans esterification, and the animal feed supplements also we can do from the remaining feed grade fats, and it can also support in the renewable energy production and livestock nutrition. So these products basically are converted from these organic waste to high valuable compounds so that the circular economy concept can be promoted and the sustainable practices can be followed. So first, acid hydrolysis of fish scale is related to collagen peptide for cosmetics. Then alkaline treatment of poultry feathers, it is related to the keratin hydroxylates for bioplastics. Then enzymatic hydrolysis of fish waste is related to fish protein hydroxylate for animal feed, and rendering of the poultry fat is related to biodiesel and animal feed supplements. Okay, let's now discuss this question. Which of the following microorganisms are majorly found in commercial buttermilk? So, in buttermilk, we have this. Basically, buttermilk is basically our low fat dairy product, which is basically a low calorie option, and it contains various micronutrients like calcium, vitamin B12, then riboflavin, and it makes the diet very nutrition and balanced. So it have the benefits of gut health, it boosts immunity, reduces lactose intolerance and inflammations, and it enhances the nutrition absorption. And it has different probiotics like Lactococcus lactis subspecies Cremoris and Leuconostoc mesenterioids. So these are the different probiotics which are there in the buttermilk. So because of these bacteria which is there in the commercial buttermilk, so the buttermilk is having this different flavor, texture, and acidity. So this Cremoris, which is Lactococcus lactis subspecies Cremoris, it is basically a lactic acid bacteria. So it produces lactic acid from the lactose, and it thicks the buttermilk and give its tangy flavor or taste because of the lactic acid. And leuconostoc mesenteroids is basically producing the carbohydrates and diacetyl from the sugar sources, and it gives the buttermilk its characteristic aroma, the buttery aroma, and the Lactobacillus bulgaricus and Streptococcus thermophilus is basically it is used in the yogurt production, not the buttermilk production. And Saccharomyces cerevisiae and Acidobacter aceti, it is basically used in the fermentation process of the alcohol and vinegar. not in the dairy. And E. coli is basically used, and this Bacillus subtilis is used in the contamination. It contaminates, not used as a dairy fermentation. So this is like the Cermaris lactis, Lactococcus lactis cermaris, and this is basically the gluconoster mesenteroids. So here, Lactococcus lactis subspecies cermaris and Leuconostoc mesenteroids are the correct answer, which are found in the milk as probiotics. Okay, so let's discuss this question. Anyone is having any doubt so far? So if anyone has doubt, please feel free to ask. Okay, so assertion is that whey is a major byproduct obtained from cheese production, which is having around 85 to 90% of the milk volume used. And reason is whey is rich in polysaccharides. It produce particularly having the amylose and amylopectin, which undergoes fermentation to produce bioethanol efficiently. Okay, let's see the solution. So here A is true, but reason is false. So this is basically the way. This watery part is the way. So whey is basically so this milk or the whey is basically having this lactose, which is from the which is the made from galactose and glucose. So this whey is basically a major byproduct of the cheese production. So it is having around 85 to 90% of the milk volume used. So it contains valuable nutrients like lactose and proteins like whey protein isolate and vitamins and minerals. So it can be useful for fermentation, animal feed and protein supplements. It is also low fat, low calorie ingredient. So it offers benefits such as high muscle recovery, improved cardiovascular health and better gut health. So we know that gym people, they usually have this whey protein, so because it is high source of protein and low source of fat to build the muscles. So the claim that whey is rich in polysaccharides is not correct, which is like amylose and amylopectin. Whey is basically having more of lactose, which is the disaccharides made from galactose and glucose. The amylopectin and amylose are basically the starch components which are found in the plants. During fermentation, this lactose, and with this is glucose, basically it is going to be fermented, and it is converted into bioethanol by lactic acid bacteria or yeast. So this route it will follow, it will glucose is converted into pyruvate, then acetaldehyde, then ethanol. So ethanol fermentation is there. So that whey is basically a good major byproduct obtained from cheese production is around 85 to 90% of the milk volume used, which is correct. But whey is not a polysaccharide or the amylose or amylopectin source. So which it is actually the lactose which is going to be converted into the bioethanol in milk or the whey. So A is correct, but R is false here. Okay, let's discuss now this, which is having column 1, the different dairy effluent treatment techniques like dissolved air flotation and then UASB reactors. Next is the wetland treatment and last is the coagulation. So in column 2, we have the functions like precipitates, colloidal solids using coagulants. Then B is removes organic pollutants using microorganisms. Then C is removes suspended solids and oils, and D is converts organic matters into the biogas. We need to match here column 1 with column 2. Anyone who would like to answer this question? So dissolved air flotation basically is a physical treatment method. So where micro bubbles which are attached to the suspended particles are there, which will causing them to float and to skimmed off and remove the oil, grease. I think there is some network problem. Okay, the network problem is resolved. Okay, so this the first technique, which is dissolved air flotation, in this the micro bubbles will be going to attach to the suspended particles so that it will become light and come over the surface. So it will float and skimmed off, uh, so that the oils, grease, suspended particles can be removed from the dairy effluents, because dairy effluent is very rich source of the fat. So what will going to happen, we can attach some, we can give, suppose this is the waste is coming, so we can give some air to it, so the air can be attached to the suspended, to this particles, suspended particles, and this can be removed separately, and underflow from this we can get, the fat will be collected here and removed, and other particles can be taken and removed. So next is the upflow anaerobic sludge blanket, which is UASB reactor. So it is basically an anaerobic treatment system which is going to break down the organic matter in the wastewater using microbial consortia, producing this biogas, which is methane plus carbon dioxide as a byproduct. So basically, this is the UASB system where uh, so this is basically the waste water will be here, and basically here the sludge bed is formed, and here is a sludge blanket will form, and basically it is having three kind of uh, three system, three or three steps is there in this. So for the down one is the sludge bed, then after that sludge blanket is there, and solid liquid gas separator is there, and here are the baffles, and here is a gas collection, biogas collection system. So at the bottom what happens, sludge granules which will having the bacteria will be at the bottom, which is sediment here. Then here in the this part, basically the turbulence generated due to the upflow of the influent and buoyant biogas bubbles as they face water and sludge resistance while moving up. So there will be some kind of resistance to the biogas. Then after that, here is the biogas bubbles formed from the fermentation. Then here are the baffles. This is the baffles. So because here the sludge will be come down and only the biogas particles can go up and collected from this part. So this treatment is effective, degrade the fat molecules to lower the COD value, and the pre-treatment such as enzymatic hydrolysis like the lipase for the fats in dairy effluents or ultrasound treatment can increase basically the efficiency of this anaerobic fermentation. Then the wetlands constructions can be used, like here the aquatic plants, soil and the microorganisms, they are going to filter and break down the organic pollutants through the natural biological process, making them an eco-friendly treatment method. So the water is purified here by filtration through these gravels or trans evaporation of the plants. So basically here is the basically the pollutant or the liquid pollutant, and here these are the plants. So here gravels, soils, and microorganisms effectively work, and they're going to produce like here they're going to give here at the bottom the filtered solution, and that will be outflowed. Then coagulation is the method where the raw water is given, and the coagulants like the chemical treatments coagulants will be there, like alum, ferric chloride. So they'll going to neutralize the charges of the colloids, causing them to form the lumps or the clumps, and uh, clump together into the flocks, and which can be easily removed through the sedimentation or filtration process. So after that it will come into the flocculent tank. So they'll be forming these lumps, and they can be easily filtered or sedimented and can be separated easily. Like this can be filtration process can be done, or effluent treatment process can be done. So here dissolved air flocculation is basically related to removal of the suspended solids and the oils. Then next is UASB reactor. So it is basically related to conversion of the organic materials into the biogas. Then wetland treatment is basically to remove the organic pollutants using microorganisms, and coagulation is precipitation of the colloid solids using coagulants. So B is the correct answer here. So let's discuss this question now. So in column 1, we have different products like biofertilizers, biosurfactants, polysaccharides, and bioplastics. In column 2, we have the respective microorganisms. So we need to match here. Okay, so let's see this solution part of this question. So there are different biotechnological methods also to treat this dairy waste. So this includes basically uh, different this dairy waste can be basically treated and can be effectively bioconverted into different resources which are very important, like it can be converted into biosurfactants, organic compounds, biofuels, bioplastic, biomass production, or biofertilizers, single cell proteins, polysaccharides, or bioactive peptides. So these can be valuable products we get from the dairy waste, so from different microorganisms. So rhizobium basically it is going to do the this is a nitrogen fixing bacteria, so that will form the symbiotic relation with the leguminous plant. It enhances the soil fertility by converting atmospheric nitrogen into ammonia and making it a key producer of the biofertilizers. Then Candida bombicola is yeast which produces sophorolipids and a kind of the biosurfactants that basically reduce the surface and the interfacial tension, making it a valuable for oil spill remediation, detergent production, and bioremediation. And Xanthomonas campestris, so it is a bacterial species which is produces the xanthan gum, a high molecular weight polysaccharides which can be used as a thickening agent in the food and industrial products due to its viscosity and stability properties. Then Ralstonia eutropha is basically synthesizing PHA, which is polyhydroxy alkonates, which is a type of biodegradable bioplastic, through the fermentation route, and making it an eco-friendly alternative to petroleum-based plastic. So here, biofertilizers basically from the rhizobium, then biosurfactants from Candida bombicola, and polysaccharides from Xanthomonas campestris, and bioplastic from Ralstonia eutropha. So, okay, so this was the last question we discussed. So the topics we discussed today are basically the linear economy, then we discussed about the circular economy, then also we discussed about the LCA, how the LCA works and what is a framework, and then we discussed about the grain or the cereals processing waste like the germ, husk, bran, and then weavers spent grains, and we discussed about how the different techniques to utilize them, like the converting them, like the anaerobic digestion to convert them into the biomass, and pyrolysis, gasification. We discussed about different thermal techniques. Then we discussed about the fruit and vegetable waste and how it is being converted into the nano-cellulose or citric acid, and the fish and meat and poultry waste, like the feathers, the blood, meal, and the scales and the meat, how they'll be used. And at the end, we discussed about the dairy industry waste utilization, where we discussed about how we treat the dairy effluent, and what are the different like techniques, and what are the different byproducts we can get from the fermentation or microbial activity. And I'll be uploading all the PPTs and in this Google Drive link, and videos on this YouTube channel link. So thank you so much for joining today's class. So if you have any doubt, please feel free to ask. Otherwise, you can leave the meeting. So please submit this assignment on time. I know there is exam, so best of luck for your exam. So prepare well and give your exams well. So okay, so I'll be uploading this. Okay, so if you have any doubt, please stay. Otherwise, you can just leave the meeting. Uh, you can just prepare for your exams, and good luck for your exams. Best wishes. So I'm stopping the recording.